Research suggests that the smell of women's tears reduces aggression in men. When someone starts crying, others often show empathy and concern. However, the biological reasons for shedding tears may go beyond simply arousing sympathy. New research has found that women's tears reduce aggression in men who are close enough to smell them. According to scientists from the Weizmann Institute of Science, human tears contain a chemical signal that reduces brain activity related to aggression. Although the study focused on women's tears, it is probably not a gender-dependent effect, their authors say. The description and results of the research were published in the journal PLOS Biology. Tears in humans do more than just moisturize and cleanse the eyes. They also appear for emotional reasons. Sadness, joy, anger, or just emotion can lead to crying. But it is not entirely clear why humans develop this ability. Scientists have shown in the past that the tears of several mammals, including mice, contain chemicals that serve as specific signals. This is an example of social chemosignaling, a process common in animals. This may also apply to humans. Studies in rodents showed that males' aggression was inhibited when they sensed females' tears. This resulted in significantly fewer fights between males. Scientists also noticed that female sand chars lubricate themselves with their own tears. According to them, females use this trick to avoid attack by dominant males. Another observation concerns young mice whose tears contain molecular signals that cause females to reject the advances of males. Emotional crying is often considered a typical human behavior. But according to Noam Sobel, co-author of the study, this is a misconception. And he gives examples. For example, dogs shed tears when they are reunited with their owners after a period of separation. Humans, like other mammals, transmit information through body odor. But until now it was unclear whether human tears would have any effect on behavior. As researchers point out, people cannot smell tears. Although most mammals have an organ responsible for detecting pheromones, in humans this organ is thought to be vestigial. The first evidence that tears could chemically influence human behavior came in 2011, when Sobel and his colleagues published their research showing that women's tears reduced testosterone levels and men's reported sexual arousal. This work raised more questions than it answered. It took years of effort to perform more thorough follow-up, in part because of the difficulty of collecting tears from donors. Scientists need at least 1 milliliter of tears to use on each experiment participant. Shani Agran, co-author of the new study, notes that using onions or another irritant to obtain tears is not an option because they are a completely different type of tears. To find out what effects people would experience after exposure to human tears, scientists used behavioral measurements, brain imaging and molecular biology. The scientists remembered about the problem with obtaining tears. So they recruited people for the study who are easily moved and can easily be made to cry. Interestingly, among the male volunteers, none were able to shed enough tears to qualify for the study. Of the approximately 100 women who signed up to participate in the project, only 6 were able to provide enough tears. Participants could induce tears in any way they liked, from listening to sad music to reading an emotional letter. However, most of them used the extensive library of sad movies. In addition to tears, researchers also collected saline drops to use in control experiments. Previous research has shown that tears lower testosterone levels in men and that this has a greater effect on aggression in men than in women. So researchers began by examining the effects of tears on men because it gave them a better chance of seeing an effect. 25 male volunteers took part in the research. They were asked to play a game designed to provoke aggression. During the game, participants were convinced that their opponent was another person. In fact, it was a computer algorithm that did everything to throw the volunteers off balance. He stole their money and resources and played against them by posing as a fraudster. In the game, volunteers could either take revenge without any benefits for themselves, or face no consequences. The researchers calculated the level of aggression based on the number of times the participant chose revenge. The team also repeated this experiment on a second group of 26 male volunteers who played the same game but inside an MRI machine, allowing data to be collected about their brain activity. All participants played the game twice and before each session were asked to smell a jar that contained either tears or saline. However, the volunteers were not told what was put under their noses. Experimental results showed that when men smelled women's tears, they were almost 44% less aggressive at play than when they smelled saline solution. The reduction in behavioral aggression was also accompanied by neural changes. Researchers observed that after exposure to women's tears, men's brains showed less activity in the prefrontal cortex and anterior insula, areas associated with aggression and decision-making. Connectivity between the anterior insula and the amygdala, an area responsible for emotional processing, also increased. Working with colleagues at Duke University, the team used molecular biology methods to test the effects of tears and saline on 62 human olfactory receptors. The researchers identified 4 receptors that responded to tears but not saline. This discovery helps answer questions about how pheromone signals are processed in humans. Agron, Sobel and their colleagues are now interested in conducting further experiments to test the effects of women's tears on other women and infant tears on adults. Sobel hypothesized that the tears of young children would likely have an effect on reducing aggression in adults. Children cannot communicate using language, but evolution may have provided them with a tool to reduce aggression, Sobel explains. We found that, like in mice, human tears contain a chemical signal that blocks aggression by male conspecifics. This contradicts the view that emotional tears are exclusively human, the authors wrote in a statement. Fossils of a half-billion-year-old predator have been discovered in Greenland. Fossils of a previously unknown group of predators have been discovered in northern Greenland. The newly discovered species was named Timorabestia copri, which means beast of fear in Latin. However, this is not about huge creatures like dinosaurs, but about marine invertebrates that could have been one of the first carnivorous animals. They appeared on Earth about 500 million years ago, were huge for their time, and were probably at the top of the food chain. The finds were made at the Sirius Passet paleontological site in northern Greenland. Timor-Beast colonized the waters near Greenland over 518 million years ago, and their discovery revealed an interesting new group of predatory invertebrates whose descendants still live today. These animals may have been some of the earliest carnivores. Timor-Beast copri were giants in their time and were near the top of the food chain. This makes them the same weight as some of the largest predators in today's oceans, such as sharks, said Dr. Jakob Winter from the University of Bristol's Department of Earth and Biological Sciences. The description and results of the research were published in the journal, Science Advances. Until now, it was believed that in the Cambrian period the dominant predators were primitive arthropods, such as representatives of Anomalocaris. But ancient ocean ecosystems, Winter points out, were quite complex, and the food chain supported several levels of predators. Timor-Beast copri are a distant relative of the sea darts, Chaetognatha, that are still alive today. However, sea darts are smaller and feed on tiny zooplankton. Their much older cousins reached over 30 centimeters in length, or about 3 times as long. They were peculiar-looking creatures with long antennae and impressive, massive jaws. They had fins on the sides of their bodies. While the 30-centimeter worm may not seem all that intimidating to us, it posed a serious threat to nearby animals in Cambrian times. Evidence of the newly discovered species' predatory lifestyle was found in its fossilized digestive system. During the analyses, scientists noticed the remains of a common, swimming arthropod of the genus Asoxes. We see that these arthropods, Asoxes, editor's note, were a food source for many other animals, said Morten Luna Nielsen, co-author of the study. They occur very often at the Sirius Passet site. These creatures had long, protective spines that faced both forwards and backwards, he added. However, these defense mechanisms apparently proved useless in the fight against the fear beast. The Timor-Beast copra ate them in huge quantities, Nielsen emphasized. New discoveries on the horizon. While arthropods appear in the fossil record around 521 to 529 million years ago, marine dart tracks can be traced back at least 538 million years. Both sea darts and the more primitive Timor-Beast copri were swimming predators. It can therefore be assumed that they were most likely predators that dominated the oceans before arthropods appeared. Their dominance lasted for about 10 to 15 million years before they were replaced by other, more successful groups, Winter said. Our discovery confirms the evolution of sea darts. Living sea darts have a separate nerve center on their abdomen called the ventral ganglion. This is a feature completely unique to these animals. We also found this in Timarabestia copri. We are very excited to discover such unique predators. During a series of expeditions to the farthest corners of northern Greenland, we collected a huge variety of animal species. Thanks to the excellent preservation of specimens at Sirius Passet, we can also reveal exciting anatomical details, including the digestive system, muscle anatomy and nervous system. In the coming years, we will have many discoveries to share that will help show what the earliest animal ecosystems looked like and evolved. Explained Tae-Yoon Park from the Korea Polar Research Institute, senior author of the paper and leader of the field expedition. New research suggests that evolution is not entirely random. New research shows that evolution may not be as unpredictable as previously thought. This may allow scientists to investigate which genes may be useful in solving real-world problems, such as the resistance of certain bacteria to antibiotics. Research published in the Proceedings of the National Academy of Sciences challenges the long-held belief in the unpredictability of evolution. Their authors suggest that the evolutionary trajectory of a genome is influenced more by its evolutionary past than by other factors. Research led by Professor James McInerney and Dr. Alan Bevan from the School of Life Sciences at the University of Nottingham and Dr. Maria Rosa Domingo Sananes from Nottingham Trent University suggest that evolution is not entirely a coincidence. And this may have big implications. The implications of this research are simply revolutionary, said Professor McInerney, the study's lead author. By showing that evolution is not as random as once thought, we have opened the door to a range of possibilities in synthetic biology, medicine and environmental sciences, he added. McInerney and colleagues analyzed the pangenome, the set of all genes in a given species, which contains sequences common to all individuals. All this to answer key questions. Is evolution predictable? Or are the evolutionary paths of genomes dependent on their history and something can be said about their future trajectory? In their research, the team used a machine learning method known as random forest. Scientists fed the algorithms with data covering 2,500 complete genomes of one bacterial species. First, scientists created gene families from each gene of each genome. This way we could compare similarities in genomes, Domingo Sananes said. Once the families were identified, the team analyzed the pattern of their presence in some genomes and their absence in others. We found that some gene families never appeared in the genome when a specific other gene family already existed. And in other cases some genes were highly dependent on the presence of another gene family, Domingo Sananes explained. As a result, scientists have discovered something like an invisible ecosystem in which genes can cooperate or compete with each other. These interactions between genes make certain aspects of evolution somewhat predictable. Moreover, we now have a tool that allows us to make such predictions, emphasized Domingo Sananes. Thanks to this work, we can begin to investigate which genes maintain, for example, the antibiotic resistance gene. Therefore, if we are trying to eliminate antibiotic resistance, we can focus not only on the focal gene, but we can also target its supporting genes, Bevan pointed out. We can use this approach to synthesize new types of genetic constructs that can be used to develop new drugs or vaccines, he added, emphasizing that this could open the door to a whole range of other discoveries. The authors of the study emphasize that the consequences of their work are far-reaching and may, for example, enable the design of synthetic genomes with an action plan for predictable manipulation of genetic material. They can also help understand the interplay between genes and identify genes that support and enable antibiotic resistance, paving the way for targeted therapies. But it is not everything. This work could help design microorganisms that capture carbon dioxide or break down pollutants, thereby helping to reduce climate change. The predictability of gene interactions could also revolutionize personalized medicine by providing new indicators of the risk of developing various diseases and the effectiveness of their treatments. Icelanders want to drill into the volcano's magma chamber. Icelandic engineers have shared a bold plan to drill into the magma chamber of the Krafla volcanic complex, which is located in the northeastern part of the island. Such a drilling could provide not only the first direct insight into the oceans of molten rock lying beneath the Earth's surface, but also enable a revolution in geothermal energy. The project is scheduled to start in 2026. Iceland is a country of volcanoes. The island is located on a mid-ocean ridge, which translates into increased volcanic activity. Thanks to this location, Icelanders have rich geothermal energy resources. Over 25% of the country's energy comes from geothermal energy. Moreover, hot water from geothermal sources is used for heating almost 90%. Buildings on the island. Iceland is dotted with thousands of deep drillings for geothermal energy. Another one will join them soon, but it will be different from the others. Icelandic engineers intend to drill into the magma chamber of the Krafla volcanic complex. In fact, there are to be 2 wells. One will allow for the first direct measurements of magma, the other will enable the extraction of geothermal energy. We will be drilling a magma chamber, says Halti Paul Ingolfsson from the Geothermal Research Cluster, Georg, in Reykjavik. This is the first journey to the center of the Earth, adds his colleague Bjorn Gudmundsson. However, the drilling will not be that deep. Some magma chambers, or underground reservoirs of molten rock, are located just a few kilometers below the Earth's surface and are within the reach of Icelandic engineers. The problem is that we usually don't know where the magma chambers are. Currently, no geophysical technique allows the location of magma reservoirs. But in this case it is different. Ingolfsson and his colleagues know where such a magma chamber is located and have bold plans for it. Krafla is one of the most active volcanoes in the world. It erupted a total of 9 times between 1975 and 1984. It was then that scientists used seismometers to determine the location of the magma chamber, about 2 kilometers under the caldera. In 2009, a geothermal drilling project conducted for the Icelandic energy company EzmCg unexpectedly hit a magma chamber near the volcano. The fact that the crew was not immediately wiped out by the volcanic eruption provides evidence that drilling into magma can be safe. In 2013, the same team that made the discovery, led by Bjarni Paulsen at EzmCg, launched the Magma Testbed (KMT) project. Scientists from the project announced their intentions to drill into the volcano to the world several years ago. According to current plans, drilling is to start in 2026, and the main goal is to deepen knowledge about magma. We have no direct knowledge of what magma chambers look like, which is obviously crucial in understanding volcanoes, said Pau Papale of the National Institute of Geophysics and Volcanology in Italy. Scientists associated with the KMT project plan to place a number of sensors in the magma that will measure the magma. We hope that we will be able to measure at least the temperature directly, which has never been done before, admitted Ingolfsson. However, the key will be to observe how the rock melts into magma, as well as to search for clues that may suggest an approaching volcanic eruption, which is extremely difficult to predict. Scientists also intend to use the well for energy research. Scientists hope to develop technology that uses the extreme heat of molten rock, thanks to which it will be possible to obtain more energy from the magma chamber than from geothermal sources. Iceland has long been a pioneer in the use of geothermal energy. The island's inhabitants use it to produce electricity and heat their homes. There are already geothermal wells in the immediate vicinity of the Crafter complex. There are a total of 33 of them, and they power a geothermal power plant that has been operating since the late 1970s. However, none of them reach the magma chamber. The drilling itself is not currently a challenge. Deeper holes were drilled in Iceland itself. Rather, the issue is what happens to the drilling equipment once it reaches the magma chamber. Magma reaches a temperature of about 1,300 degrees Celsius. Gudmundsson stressed that tools must be developed that can withstand the extreme conditions in the magma chamber. The same applies to sensors that are supposed to measure magma. They must withstand enormous pressure and extreme temperatures.